Hello, everyone. I am Nam, a PhD candidate from Daifeng Guang Lab at the University of Wisconsin-Madison. It is my pleasure today to present our work on a deep learning model called Varmole, a bio-drop connect model for prioritizing disease risk, variants, and genes. So I'll start with the motivation. As you may know, genome-wide association studies found many genetic variants associated with diseases. As you can see in this Manhattan plot, among various coordinates in this chromosome, it's like SNP2 and SNP1 have a significant association with a disease. However, the detailed molecular mechanism from these associations are still unclear. On the other hand, if we can combine different multi-omics like genomics, proteomics, transcriptomics, to get a big picture, it may help us to understand the molecular mechanism in a detail, like gene regulation or functional genomics. So the question is how to integrate multi-omics and also find causal variants and genes and networks for disease. So to answer for the question of multi-omics integration, there are a bunch of methods called multiview learning. And actually, we did a systematic review about that topic on PLOS computational biology. So here I will present some essences. Looking at the figure in the center here, it is biological mechanisms. It is the result of the interaction both within each omic and also the interaction across various omics. So by using traditional or single view learning, we can only learn the interaction pattern within each omic separately, depicted by the function f here, like f1, f2, f3 for 3 different omics. But using multi-view learning, we can also learn the crosstalk patterns or the interaction pattern across different omics, depicted by the omega core term here, which is actually the core regularization between the 2 function f. So in this pairwise fashion, we can learn, for example, how SNPs can break transcription factor binding sites between the genomics and proteomics, or how transcription factors control gene expression between proteomics and transcriptomics. So Varmole is a search multi-view method, which is also an interpretable architecture. So its architecture consists of 2 main parts. The first part resembles the mechanism of how variants and gene regulation giving rise to the gene expression. And the second part is like resemble the mechanism of how gene expression giving rise to the phenotype. First part, it is the first layer also, and it's a transparent layer which take the input from 2 different views, variants X and gene Y. And it also embedded the network A1, which is a gene regulatory network, and the network A2, which is the EQTL, as the prior knowledge to embed it in the architecture. So gene regulatory network is like linking transcription factor to the target genes, and the EQTL here, which links the SNP to the expressed genes. So, as I said, this like resemble how variants and gene regulation giving rise to gene expression. So in this layer, the number of the node here is equal to the number of the gene here. And for the second part, it is contains of multiple fully connected hidden layer. And for disease classification, we use cross entropy so that the full optimization problem a Varmole is specified by this. And we train Varmole with lasso regularization to deal with the problem of high dimension and low sample, which is very common amongst biological data sets. So how we embedded the prior knowledge of gene networks into the architecture. We are inspired by drop connect regularization, which is a regularization technique similar to dropout. So in training a neural network, if we drop out some hidden node like this randomly in training, it's called dropout. And instead, like if we don't train the network with the full connection depicted by the parameter W here, but like I want to randomly drop out some connections, it called drop connect. So these are two very simple but effective regularization techniques. And in Varmole, we did the like similar thing to drop connect, but instead of drop the connection randomly by taking the element-wise product between parameter W and a random matrix, we use the GRN or EQTL as the drop connect mask to drop those connections that are not in the EQTL or GRN. So the second aim of Varmole is interpretation, how to prioritize genes and SNPs and the links between them. We employed a method called integrated gradient to do such thing. So the idea is given a model F, an input X, and the output Fx of that specific input, we can calculate the attribution or the importance scores of each feature I of that input X as the integrated gradient. So we can, in Varmole, we can calculate the importance scores of each SNP or each genes, or we can extend the method to calculate both the link importance between two genes or between a gene and a SNP. And this is very important for the downstream analysis, such as enrichment analysis. So we applied Varmole for the classification of schizophrenia sample. The data set we use here is RNA-seq gene expression genotype data for about 500 schizophrenia versus about 900 non-schizophrenia human brain samples. We also embedded the GTEx, EQTL, and psychoencode gene regulatory network for human brain front cortex. And after filtering out SNPs and genes that match those in the EQTL and GRN, we come up with like 100,000 SNPs and 2,500 genes. So training this data set with Varmole, we get the high accuracy score, as you see in this bar chart, compared to the other state-of-the-art method. So you see Varmole is here. Okay. And to reveal the biological meaning, we did some enrichment analysis to find the prioritized gene functions and regulatory links for schizophrenia. So as you can see, this is a list of enriched functions, and this list is supported by many previous studies on schizophrenia. Those functions includes like neuron development, axon guidance, scent adhesion, etc. So we also find the SNP gene pairs on the interacting enhancers and promoters have significantly higher importance scores. And also potential regulatory roles of prioritized SNPs to genes via enhancer. So Varmole can be extended in many different ways, like we can integrate the single cell data and embedded cell type gene regulatory networks into a deep neural network model. We also can add more additional omics like epigenomics, ATAC-seq, to learning the interactions among enhancers or promoters. And of course, deeper phenotypes like neuroimaging or behavior can be used. So that's all of my presentation. We appreciate the support by NIH grant for this work. Thank you for your attention, and I'm happy to answer any question you may have.
Hi, everyone. Thank you very much for the nice talk. I think we can have a couple of questions. We already have one from the audience. So the question is, what would be your strategy for a condition like NF1, where the phenotype is highly variable depending on the mutation site?
Yeah, thanks for the question, but I don't know what do you mean by NF1 here? The phenotype highly variable depending on the mutation site.
Well, I guess the phenotype is maybe in NF1 is probably you could put it as a placeholder, but if you have a phenotype that is highly variable depending on the mutation site, okay, so it is neurofibromatosis.
Oh, I see. So our model here is like to combine genotype and gene expression to classify the phenotype. So
So is it sensitive to certain types of phenotypes, or do you think that it is very generalizable to different types of phenotypes?
Yeah, right. So our model is very general, can be generalization. So I don't think it depends on the phenotype depending on the mutation site here, because
Yeah, I have a question. Could you explain a little bit by accuracy of a model? So what is truth here? Because you don't know what are the regulatory variants for different diseases. So what is it that what is your accuracy metric actually assessing?
So I use the balanced accuracy score because in our data, the data is quite imbalanced. And we compare the Varmole with other state-of-the-art method like AdaBoost or KNN and the DSPN with the same data set.
I guess my question was like what you need a ground truth of what is a true variant versus not. And I think that I was just wondering where it came from, right? I mean, your model is trying to predict which variants are causal for a particular phenotype. So those must come from somewhere.
Yeah, we have a truth label. Like we have like about 500 schizophrenia versus about 900 non-schizophrenia human brain sample from the front cortex.
All right. I think thanks for the presentation. I think maybe we can move on to the next speaker. There's another comment from Lars Eriksson. Maybe you guys should just meet up after the presentation, after this session. But let's move on to the next talk.